Welcome to the lecture on the first day of Chapter 9. We're going to talk about the eye and vision in the next two chapters, actually. These are real interesting couple of chapters. We're going to see that our eyes and our brain that's involved in vision work in some really interesting and surprising and often complicated ways to produce the things that we see and perceive. And we're going to find also that we're going to see a lot of similarities to what we saw with taste and smell. But we're also going to find some fairly big differences in how signals from light are transduced in our eyes and processed in the brain. There's going to be a lot more involved because vision is a lot more complicated than smell. For example, think about vision. You could think of the different colors that you see as the different smells that you might be able to detect in the environment. But what else do we have in vision that we don't have in smell? In vision, we also have movement. You can't really smell if something's moving. In vision, we have shapes. You can't really tell what shape things are. In vision, we have borders and lights and dark. You really can't smell where the border to something is or where we have more or less smell even. You can move your head around and try to detect where smell is coming from, but we really have a hard time getting directionality from our sense of smell, and tastes in a similar way. In terms of the speed at which something is moving and the direction it's moving, we need to detect that with our vision, and that's something we can't detect with smell. So there are a lot of additional things that our visual system has to detect that the smell and our taste system don't. And so we're going to see that it's more complicated and involve a lot more processing in the brain than we saw with smell and with taste. So let's get into this and start talking about light, because that's what enters our eyes and that's what we are seeing. So whenever you see anything, it's because light is bouncing off of that thing and entering into your eye. And then your eye picks up that light. Even dark things that you're looking at, unless they're completely black, would have some light bouncing off of them into your eye. And so what is light, and how does our eye transduce that light into neural signals? So let's look at light. Light is a form of electromagnetic radiation. You can think of electromagnetic radiation as little packets of energy that fly through the air. And when they go through the air, they move in a wave pattern as they move through the air. So light is really kind of a combination of energy and matter. And people still study how light works and what it's made of. But essentially, these little packets of energy are called photons, and they travel through the air. Now, there are other types of electromagnetic radiation that can go through the air, like X-rays or infrared rays or ultraviolet rays. But when we talk about light in this class, and for vision, we're talking about visible light that we can detect with our eyes. There's a lot of other electromagnetic radiation going on, like I just mentioned, radio waves, for example, that also contain these small packets of energy that travel through the air in waves, but we can't see those. Our eyes don't detect those. There might be other animals that can detect those other types of light. For example, I know butterflies can detect ultraviolet light, but for the most part, when we talk about light here, we're talking about visible light. But it is on a spectrum of electromagnetic radiation. When light travels through the air, the amount of waves that it goes through as it travels is called the frequency. So if you look at this wave of light traveling here through the air, this light is green in color, and it has a particular wavelength, which means the distance between two troughs or two peaks of this wave. Again, it's traveling through the air in this wave-like pattern. You might have another color like blue or purple, let's do purple, that would also travel through the air in waves, but its wavelengths would be much shorter. And I haven't drawn that very nicely, but the different wavelengths determine the color of the light and also the energy of the light. The energy of the light would be how much energy the particles, the photons, are carrying. So actually, ultraviolet light or purple light, because it has very short wavelength, has a lot of energy. That's why ultraviolet light from the sun can destroy your DNA, whereas the blue or the red and the orange light from the sun is not as damaging. Really, when we put on sunscreen, we're trying to block the ultraviolet light. It's the higher energy light. Those packets can crash into our DNA. Those packets of energy can crash into the DNA in our skin and mutate the DNA because they have a lot of energy, so they can knock things out of place. Colors down towards the red end of the spectrum have much broader wavelengths. So wavelength is a property of the wave of the electromagnetic radiation. It's also correlated with the color of the light and also correlated with the energy of the light. So these guys with big wavelengths have low energy, and these with short wavelengths have high energy. Amplitude is a measure of the brightness of the light. So if that wave is traveling in big, if the waves are really high, if the peaks are very high, then that means that light has a lot of amplitude. It means there's a lot of light there. It's a very bright light. The energy is proportional to the frequency, and the frequency and the wavelength are directly correlated. So the shorter the wavelength, the higher the frequency. I should say they're indirectly coordinated. The shorter the wavelength, the higher the frequency. And so, and also the higher the energy. So short wavelengths have a high frequency, long wavelengths have low frequency and lower energy. And this is just showing you the other electromagnetic radiation that flies through the air. We've got gamma rays. Those are radioactive rays. We've got X-rays, ultraviolet rays. These all have a lot more energy than visible light. Visible light just fits into this little narrow part of the big electromagnetic spectrum. And so our eyes are designed to pick up only that electromagnetic radiation. It's possible that you could create some sort of sensor or maybe even another type of eye that could pick up infrared rays or that could pick up ultraviolet rays. And in that case, they would see more than we see. They would see things that we don't see. When light interacts with something, it does a number of different things depending on what that thing is that it's interacting with. If it's hitting a mirror or some reflective surface like glass, it will be reflected. So those light packets of energy will come in and bounce off of the surface. Other surfaces that are not as reflective might absorb the light to different degrees. Black colored things are going to absorb light better than white colored things, because whatever that thing is, that black thing has a molecular structure that absorbs the energy well. And that's why something black, like blacktop sitting out in the sun, can get very hot, whereas something whiter like concrete doesn't get as hot. It's the molecular structure of the substance that determines how well it absorbs those photons of light. In other cases, like in the lens of our eye, the light might be refracted. And that just means that the light is bent. It's traveling in a straight line, and then when it hits that surface, it bends. But it also keeps going. through that surface. So the interface between air and our eye, there's a difference in the composition of the air and the composition of our eye. And whenever you have an interface between two different media like air and eye, which is mostly water, then the light will bend. You've all seen this when you go into the swimming pool and you look down at your feet. Your feet don't look normal. They look like they're extended out or bent. That's because light coming from them, when it leaves the water, is refracted and bent. And so it comes at your eye in a slant that it normally wouldn't come at. And so your whatever's underwater looks strange. I'm not going to spend any time on the anatomy of the eye because you get all of that in lab. So you will need to know these structures for the exam, but we'll let the lab take care of those structures. Just briefly, I want to mention the blind spot and the macula fovea. So we have blood vessels and nerve axons that come into our eye and perfuse and innervate our retina, the inside of our eye. Those blood vessels need to they need to have a place where they exit the eye, and the axons need a place where they exit the eye too. And that's the place that they do that is the same place, and it's called the optic disc. And so there is a place in both of your eyes where blood vessels and neurons can enter the eye or exit, depending on whether they're arteries or veins, and also nerves, depending on whether they're afferent or efferent nerves. And these nerves enter the eye through that optic disc, and the blood vessels do. And so at that point in your eye, there aren't any photoreceptors. And so light hitting that doesn't trigger any sort of response in neurons because there aren't any actual neurons there. There's just the axons. And the axons are not responsive to light. So that little place in your eye is called the optic disc, and it's a blind spot. And you're going to do an experiment, or you have in lab, where you look for that blind spot. In your book, there's a little activity that you can do too, that you can see where your blind spot is in both eyes. The macula is what we call, is the part of the eye that's in the center of your eye, and it's going to be involved in picking up things in the central part of our visual field. We'll talk about later about visual field, but if you're looking straight at something, then the center part of what you're looking at, where you can focus very well on things, is called the central part of your vision, the central visual area. And the outside part of our vision, what we normally think of as the periphery, is called our peripheral vision. The macula then picks up light that from things that we are looking directly at. And the fovea is the very center of that visual field. Things that are in the very center of what we're looking at. And that's the place where we can focus best and see things with the greatest amount of acuity, which is sharpness. Let's take a look at how light and images are focused onto the back of our eye, our retina. What we will see is that as light goes through our eye, the first thing that it hits is the cornea. The cornea is a very clear tissue on the very front of our eye. It's actually a connective tissue, and it's quite a thin layer, but it allows light to pass through it. But because it is curved, it will refract or bend light as the light travels through. The cornea is actually going to bend light. Of all the eye structures, the cornea will bend the light the most as it travels through. And when it bends that light, it will focus it onto the back of our eye. What do we mean by focus it? Let's take a look at what happens when light strikes our cornea, which is a curved surface. Let's take an example of a tree here. And so light from the sun hits the tree, and the light is going to bounce off of that tree. Some of it will be absorbed. Of course, Some of the light will be absorbed by the leaves as they do photosynthesis, but some of that light will be reflected off of the leaves and off of the tree and bounce off in many different directions. In fact, let's take the top of this tree here. Light will hit the top of that tree and bounce off in lots of different directions. Some of that light will travel towards our eye, but a lot of the other light from that tree will bounce off in other different directions. Same thing for light off the trunk. Light off the trunk is going to bounce off of the trunk in many different directions. Some of that light will reach our eye. Let's look at the light rays then that reach our eye. So I'm going to take a light ray from the trunk, and this light ray is going to come, and there's going to actually be a lot of light rays that come off of this trunk. But let's take this light ray here that's going to come off the trunk and travel in this direction to our eye. It is hitting the cornea at the very bottom of our cornea. So there's a very shallow angle there that it's hitting it at. When it hits that light, it will turn towards the center of the eye. So when light hits, light that had been traveling in air hits something that is mostly made of water, it will bend. It will slow down. And when it does so, it will move in a direction that we say is perpendicular to the surface here. So if I draw a perpendicular line, if I draw a tangential line that runs right along the surface of the cornea like that, then what I'm going to see is that the light from this tree that was traveling through the air is going to turn to a direction that's perpendicular to the surface of the cornea. And so it's going to travel in that direction. So I haven't drawn it perfectly perpendicular here. This isn't quite a right angle because it depending on where it hits, it's not going to be exactly perpendicular. But that light ray will come into the cornea like that. Now let's take another light ray and let's send it from the bottom of the trunk towards the center of the cornea. Whoops, I wanted that to be red. Okay, so another light ray is going to bounce off the center, and let's put a perpendicular line there. Okay, a tangential line. And then we're going to see that the light ray will continue on perpendicular to that blue line that I just drew, almost perpendicular to it, and cross that first beam at that location. So this is where they're crossing. This is called the focal point. Now let's take a third line and send it up. It bounces off the trunk of the tree, but actually bounces up to the top of our eye. Okay, let's draw a little perpendicular line there. I keep saying perpendicular. That's the tangential line, and now we'll draw a perpendicular line. So light from that tree trunk will hit our eye in many different places, but wherever it hits our eye, it will be refracted or bent, and those light rays will then converge at one point that we call the focal point. Now if we take another line from the top of that tree trunk, we'll make this one green. So light bounces off the top of the tree trunk, hits here. So it's now hitting at a different angle than this light beam was, right here. This is hitting at a different angle. And so it will be refracted a little bit more. Because this is hitting the eye at, this light beam here is hitting this eye, but it's not hitting it at as steep of an angle as this light ray was, so it won't be refracted as much, but it will be refracted down. And then let's take another beam of light that comes off and hits the center of the eye. Okay, it'll be refracted also down, but not as much. And then another beam of light that's going to come in here is going to hit it at a very steep angle, and so it'll be refracted quite a bit. But all of those beams of light from that top of that tree will end up focused in the same place. And this is the focal point. And the reason that they end up getting focused or converging in the same place is because of the curvature of that cornea. So the curvature of the cornea helps that light bend into one place. And this is the focal point. Now the distance between the front of the cornea and the location of the focal point is what we call the focal distance. And the more curvature the cornea has, the shorter the focal distance is. So let's take another cornea that's not as spherical. And what we'll see is when light comes in, we'll just do the trunk of the tree here. When light comes in down here, it just is bent a little bit because the cornea is not as spherical, and so what we see is that the focal point is much further back in the eye. And so depending on how spherical your cornea is, you may focus light at a different place. And depending on where your retina is, let's draw the retina in orange. If the retina is right here, if the retina is right here, then the light is focusing right at the back of your retina, and images will appear sharp to you. You'll have a lot of visual acuity or sharpness. But if this were where the back of your eye were, if this was where the back of your eye was, then those light beams are not converging at the back of your eye. They're converging beyond the back of your eye. Well, they're not going to reach there because the back of the eye would block them, but they would have converged there if light had been able to keep going. And so that the trunk of that tree is going to appear out of focus because the light is coming in and hitting your retina at different places instead of at the same place. And so your brain is going to detect that as multiple things instead of one thing. And so you'll see fuzziness. If you wear glasses, you know what I'm talking about. You know, if you think about looking at something without your glasses, it's fuzzy, like there's two or more images that are offset from each other, and you can't see them. That's because that light is converging at different places on your eye. So some of us have a long eye. Some of us have an eye, a cornea shape and an eye that's fairly long. And what ends up happening is that light converges before the back of our eye, and so we don't see anything. We don't see clearly. And some of us have really short eyes, and so the light would have converged beyond the back of our eye, but on the retina itself it's not converged, and so we don't see clearly either. These two types of eyes are what gives us nearsightedness and farsightedness. So most of you probably, or a big chunk of you probably have to wear corrective lenses, and you may either be nearsighted or farsighted. I wanted to show you how this works. So if somebody is nearsighted, that means they can see things that are close up, but they have a hard time seeing things that are far away. Having nearsightedness is also called myopia, and it arises, one of the main reasons that it can arise is because your eyeball is too long. Hyperopia is farsightedness, and this comes from your eyeball being too short. All right, let's look at somebody who's nearsighted, who has an elongated eye, and see why things are clear to them up close, but are fuzzy to them when they're further away. So let's take a tree that's really close. Okay, and let's say we're looking at the top of the tree here. So light's bouncing off of that top of the tree in many different directions. But because it's so close, the light from that tree will hit a large portion of our eye because our visual field is taken up more by that tree than if it were further away, right? If it's right up next to us, then that's about all we can see is the tree. But if it's further away, we can see a lot of other things too. But let's say it's really close, then let's just take some light that's coming off at the top of the tree and see how it will hit our eye. So it's going to hit a large portion of our eye. It's going to hit the top of our cornea here. It's going to hit the middle of our cornea, and it'll even hit the bottom of our cornea. It's going to hit a large portion of our eye. These light rays, when they hit the cornea, then will be refracted in a direction that's perpendicular to the surface of the cornea. So this one won't actually be refracted that much. It'll continue almost straight down. This one is going to be refracted like this. And this bottom light ray will be refracted like that. Okay. Yeah, I haven't drawn that bottom one very well. Let's be a little more careful with that bottom one. So that light ray will hit the bottom of our eye right there. Okay, and perpendicular to that is going to be like that. Basically, those three light rays from the top of the tree will hit the same part of the back of our eye. And so because they all hit the same place, our eye will perceive that as one point of light, and so it won't look fuzzy to us. It'll look sharp. We'll have acuity there. Okay, now let's look at a tree that's further away. Because it's further away, of course, I've drawn it smaller here. Let's watch and see what light rays do when they come in from the top of this tree. Okay, the light ray will come in from the top of the tree, and it will strike the top part of our cornea there. And because it's coming in at a steeper angle than this light ray was here, it's not going to be able to be refracted as much. It would like to travel in a line that's perpendicular to this line that I just drew here, but because it's coming in at a steeper angle, it won't be able to refract as much, and so it'll go like that. Then let's send another line to the middle of our cornea. Another light ray comes here. Whoops, I should try to draw it as straight as I can. Another light ray comes from the top of the tree and hits the middle of our cornea, and it's coming in pretty straight, and so it won't be refracted or bent that much. Okay, and then this third one comes in and it'll hit the bottom of our cornea. Whoops, I can't seem to draw a straight line. Hit the bottom of our cornea. Okay, and it's coming in at not much of an angle either, so it will be refracted, but not much. And you know, I may have fudged the lines a little bit, but what I'm trying to show you is that the focal point from those lights that came from the tree that hit different parts of our cornea will converge they'll converge in front of our retina. And so by the time they get to our retina, they will be hitting different parts of our retina, and so the tree will look fuzzy to us because it's far away. That myopia can be corrected by putting a concave lens in front of your eye, and that's what your glasses would do, that bend the light outwards a little bit. What that concave lens will do is bend the light outward so that when the light converges, since it was bent outward at first, it'll bend the light outward like this, and since it's bent outward like that, to converge, it's going to converge at a place further back because it started spread out, and so to get it to converge, it's going to have to be further back on your eye. So if you have two lines of light here that come in and converge, with the cornea here, without any corrective lens, they might converge here, but if you have a corrective lens that bends the light outward like this, like a concave lens would do, then the convergence won't happen until further back in the eye. And hopefully your doctor has set it up so that light is bent in such a way that it converges right at the back of your retina, and so that image will then look sharp again. Let's look at hyperopia now. This is where you are farsighted, so you can see things that are far away fairly well, but things that are close you can't. And this arises when you have a shorter than normal eyeball. Let's put that tree there again then, a tree that's very close up, and remind ourselves that when we had that tree close up here above, the light from the top of that tree converged in one place on the back of our eye, and that tree was in focus. Notice that it's focusing back here. If we had the same corrective power or the same refractive power in our cornea in this bottom eye, then those beams of light would converge back here behind our eye. And so our retina would actually pick up an unfocused image. So to correct that, what we need to do is put a convex lens in front of the eye, and what that will do is it will cause the light to converge before the back of the eye, because what it'll do is bend the light in. So let me draw this in blue. So I'll put a convex lens here, and what that will do is it'll bring this light in, bend this light in such a way that these 3 points of light now converge on the back of the eye instead of behind the eye. They basically cause more, the convex lens here will cause more refraction, which is what we need, because we need the light to be bent more so that it converges at the back of our eye. It's not bending enough, and so it's not converging until beyond the back of our eye, which of course it never gets to because the eye would block it. But the retina is getting unfocused light. And so by putting that convex lens in there, we refract the light more before it hits our cornea, and so then our cornea doesn't need to refract it as much to get it to focus on the back of our eye. Our lens, which is also a part of our eye that can provide refractive power, can also bend light. And when we look at something up close, the ciliary muscles in our iris will relax. They'll contract. I'm saying it wrong. They'll contract, and by doing so, they'll come in closer to each other, and so the ring around the lens will get smaller, and that will allow the lens to ball up into a more spherical shape, and that's called accommodation. And this is what our lens does when we look at something close up. We need more refractive power when we look at something close up, and so the lens will relax into a more spherical shape in accommodation to do that. When we're looking at something from far away, the light rays are coming in more parallel to each other, and so we don't need as much refractive power. The cornea is sufficient to focus those on the back of the eye. But with something that's close up, we need a lot of refractive power to bring that image into focus on the back of our eye, and so the lens will ball up or accommodate to help refract that light. When you go into a bright room, you can see your pupils constrict. This is called the pupillary light reflex, and what happens is you've got light that stimulates neurons in your retina that we'll learn about here, ganglion cells, and some of them will send axons to the brainstem, and in the brainstem we find motor neurons. So let's just, ganglion cells in our retina will receive input from our photoreceptors, which are receiving input from light, but let's just stick with ganglion cells now. These are the neurons in our retina, and they will send information about the light that we're encountering to our brainstem, specifically the midbrain. And in the midbrain, we have motor neurons that will efferently innervate the pupillary, the iris muscle, and cause it to contract. And so this reflex happens, and this information from the brainstem will be sent to both eyes, and so both pupils will contract at the same time. Even if you were to close one eye and shine bright light into just one eye, both pupils contract. This is what we call consensual. And that will adjust the amount of light that can come in. Of course, when we go into a dark room, our pupils will dilate because this light reflex also will allow those muscles to relax. Another really important concept that you need to understand in this chapter is something called the visual field. It's really simple though. The visual field is everything that we can see. And so we have, if we're staring straight ahead, then of course we could divide our world into in quadrants, right? We could say we have the upper part of our world, the lower part of our world, the left part of our world, and the right part of our world. And so we call that our superior visual field, our inferior visual field. And then we'll learn later about the central visual field and then the peripheral visual field. Okay, now let's get into the retina and talk about the cells that are in there that help us detect light. There are 4 types of cells that we're going to learn about: photoreceptors, bipolar cells, ganglion cells, and horizontal cells. There are also cells called amacrine cells in the retina. They are going to be involved. They're important also and involved in visual processing, but we don't know exactly what they do, and so we can't describe their function as well. So we'll stick with those other 4. What happens when we see? Light is going to come into our eyes, and it's going to affect the photoreceptors. Now, the photoreceptors are actually at the very back of our retina. So when light comes in, it actually will pass through the ganglion cell layer and the bipolar cell layer, and it'll pass through part of the photoreceptor, and it will actually have its effect here on the outer segment of the photoreceptor. Light then will affect that, and the photoreceptor will change its membrane potential and release more or less neurotransmitter. That neurotransmitter will affect the bipolar cell, and the bipolar cell will then create a graded potential and release more or less neurotransmitter depending on whether it's depolarized or hyperpolarized, and that will cause the ganglion cell to either depolarize or hyperpolarize, and if it depolarizes enough, it will fire an action potential and send information to our brain. This means then that the information about our visual field is transmitted in a certain order from photoreceptors to bipolar cells to ganglion cells onto our brain. And so the eye, the retina, is actually a little bit inside out if you think of it. And this is one of the first things that kind of throws students, is that the light from our world passes into our retina, and it actually bypasses the ganglion cells and the bipolar cells to reach our photoreceptors. And this might seem a little strange. Why would the eye be designed in such a way that it has to get through these other layers of cells before it actually binds to the light receptive cells, which are the photoreceptors, the rods and the cones? Well, we're going to see that the rods and the cones need to be towards the back of the eye for a specific reason. And so the setup is helping the photoreceptors stay healthy. Right at the very back of the retina is a dark tissue called the retinal pigmented epithelium. And this retinal pigment epithelium is going to be involved in helping the outer segments of the photoreceptors be regenerated. What we find in the outer segments of these photoreceptors is a pigment, which is like a colored protein, and this pigment in our photoreceptors called rhodopsin or conopsin, they're depending on whether we're talking about rods or cones, In rods it's called rhodopsin, and in cones it's called conopsin, but it's this pigment protein that can capture light. When it captures light, it needs to be, it's basically taken offline, and it needs to be regenerated. And that retinal pigment epithelium helps regenerate that pigment. When those rods and cones then are placed back at the back of the retina, they're right next to that retinal pigment epithelium, which helps regenerate those outer segments of the rods and cones. You can think of those outer segments as kind of like dendrites for a neuron. And then you have the cell body here, and then you have the axon here, which synapses with the bipolar cells in the outer plexiform layer. The cell bodies of the photoreceptors are called that layer is called the outer nuclear layer. So these are photoreceptors, and we have two kinds, the rods and the cones. And then the bipolar cells form the inner nuclear layer, and they synapse at the inner plexiform layer with the ganglion cell layer. Now these cells are the neurons. They're all neurons, but these are the neurons that transmit the information to the brain. Let's look at these rods and cones in a little more detail. Rods and cones are special because what they will do is something called transduction, phototransduction. And in phototransduction, what we do is we convert electromagnetic radiation into neural signals. In this case, we're going to be converting it to the release of neurotransmitter. What we'll see with the rods and the cones is that as light strikes them, they will actually hyperpolarize and release less neurotransmitter. And I know that seems a little strange, but light is actually going to turn them off. And relative darkness will cause them to release more neurotransmitter. But it doesn't matter whether light causes them to release more or less neurotransmitter. Really, that's just the way they're set up, is that they release less neurotransmitter when light strikes them. The important thing is that they transduce or convert a signal from an electromagnetic radiation form to a chemical form, the release of glutamate. The pigments that can absorb the light are found here in the outer segments. Cones have much smaller outer segments, and they're tapered at the end. And so they're not as sensitive to light. To activate a cone, or to inactivate a cone, I guess, you need more light. Whereas because a rod has a much bigger antenna, it's much more sensitive to light. And so just a little bit of light can hyperpolarize a rod, whereas you need more light to hyperpolarize a cone. The pigments in rods are sensitive to most all wavelengths of light. They're most sensitive to green light, but they can absorb any wavelength of light. And that pigment in rods is called rhodopsin. In cones, they have a pigment too called conopsin. And in fact, cones have three different types, can have three different types of conopsins. They can have a red, a blue, or a green conopsin. And what that means is that those opsins, each of those three opsins responds or absorbs a different color of light better. Green conopsins absorb green light better than the other two. And so depending on what kind of cone you are, whether you're a red, green, or a blue cone, you respond better to different colors of light. And so cones are responsive to different colors, whereas rods are responsive to all colors. Humans have a duplex retina, meaning that we have rods and cones. But the rods and cones are not distributed evenly on the back of our eye in our retina. We find that in the macula and the fovea especially, we have a very, very high concentration of cones and almost no rods. And in the periphery of our retina, we see a lot of rods, a much higher concentration of rods than cones. That's what this graph is showing here. So if this is the very center of our fovea, we see that at the very center we have a high concentration of cones. And as we move away from the fovea to the periphery of our retina, so this would be we have the center of our retina, which receives light from objects that we're focused on. And then the periphery of our retina receives light from objects that we're not focused on. So right now I'm looking at the camera, and so light from the camera is hitting my fovea, but light from my monitor, my keyboard, and the rest of my office are hitting the peripheral part of my retina. There are cones out there, but they are at low concentrations out there in the periphery, and I have a lot of rods. Now I want you to go home and do an experiment tonight. I want you to go into a dark room and wait for your eyes to adjust. And then I want you to, once your eyes have completely adjusted after maybe 10 minutes or so, I want you to just start looking around your room. And I want you to notice that when you focus on something in a very dark room, that thing that you're focusing on disappears. So maybe you just focus on the corner of your closet, and you'll notice that that part disappears. Or maybe you have a tiny beam of light somewhere in your room that's really dim, like maybe your phone charger or something like that. If you look at it, and it's a small enough light, when you look directly at it, it can disappear. When you look at stars, if you try to look directly at a dim star, you'll notice that it disappears too. This is not because of your blind spot. It's because that dim light coming from the corner of your closet or from your phone charger or from a dim star is hitting your fovea, and because you pretty much only have cones there, and those cones aren't very sensitive to light, they don't pick it up, and so you don't see anything. But if you turn your eyes just a little bit away, that light from the corner of your closet or from a dim star hits the periphery of your eye, because now your fovea is focused on something else. And so that light from the star hits the periphery of your eye, and you have a lot of rods there that are very sensitive, and so they can pick up that light, and they can activate and trigger a signal and tell you that you're seeing something. So try that when you go home. Try experimenting with your fovea and see how insensitive it is to low levels of light. The advantage of the fovea, though, is that it can give us a lot of visual acuity, because what the fovea does is it packs in a lot of cones, and each cone can pick up a different photon coming from something. And so what happens is each cone can receive input from a very small part of your visual field, and it can distinguish it from another closely adjacent part of your visual field. So let's say there are two dots written up on the board, and you look at them. If you look at them with your fovea, you'll be able to distinguish that they're two dots. But it may be that if you look at them in your peripheral vision, you might not notice that they're two dots. Try this experiment. Try looking straight at a word on your page, and then while still looking at that word, read the words that are further down on the page. You can't do it. That's because your periphery is receiving light from the bottom part of your page, and that's just rods and cones that are spread out. But in your fovea, those cones are packed in, and so they can see that light. They can see, they can give you a lot more acuity, because they can distinguish between 2 points easier. And being able to distinguish between 2 points is the definition of acuity. In the fovea, then, we have each cone can receive information from one point of light. So there'd be another cone here that's connected to a bipolar cell here, that's connected to a ganglion cell here. And so if you have two close points of light that you're looking directly at, they'll hit two different cones in your fovea. And your ganglion cells, there will be two ganglion cells that are activated. Okay, let's look at the periphery here. why can't, if I'm staring directly at a word on my page, why can't I read words that are further down on the page without looking at them? That's because that light is coming, the light from further down on the page is coming in and hitting my peripheral retina. And because I don't have a one-to-one correlation between cone, bipolar cell, and ganglion cell in the periphery like I do in the fovea, I'm not going to have as much acuity. So remember, these 2 points of light in my fovea activated 2 different ganglion cells. What about if I'm going to make this a cone here. I'm going to make this other guy a cone instead of a rod. Let's have 2 cones that are next to each other in the peripheral retina receive input from 2 closely opposed points of light. And so these 2 cones will be hyperpolarized. That will cause these bipolar cells to activate, but they all converge onto one ganglion cell. And so now there's only one action potential traveling to my brain instead of 2. And so my brain will perceive these 2 points of light as a single point of light. That means I have lost acuity because 2 points of light appear as one when there really are 2 there. So our peripheral retina has much less acuity than our central retina. And this is because of convergence. It's actually twofold. The first reason is that multiple photoreceptors converge onto one ganglion cell. The other is that the density of cones and rods actually is low the further we get out into the periphery of our retina. So things that are in your most peripheral part of your visual field, they're not hitting the center of your eye. They're hitting the most peripheral parts of your retina. So if this is your fovea here, this is receiving input from things that you're looking directly at. Out here, you're receiving input from things that are in the periphery of your vision. And you have the least acuity out there. You do have cones because, of course, you can see color. But even then, try looking straight at something and then telling me what all of the colors are of everything that you see in your periphery. I'm right now looking at the camera, and I can tell that there's something hanging on my desk over here that has a color, but I can't quite tell what it is. It's a really strange phenomenon if I sit there and think about it. I think it might be orange. It's more like brown. But I couldn't tell that because I don't have a lot of cones in my periphery. I don't have a sufficient density of cones in order to tell what the color is. I forgot another thing that your book mentioned, which are the terms scotopic, photopic, and mesopic. This refers to the level of ambient light that there is. In scotopic conditions, this is like going into the dark room and letting your eyes adjust. Have you ever noticed that when it's really dark, you can't tell colors anymore? That's because your cones basically are not picking up enough light to activate, and so you can't distinguish colors. The only thing you just see black and white, basically, or black and gray. You can't really distinguish colors. Rods, remember, pick up all wavelengths of light, and so and they're the only ones sensitive enough to pick up light in scotopic conditions. There's just not enough photons hitting your eye to activate the cones. And so you can see things, but you can't see them very clearly because you don't have a lot of rods in your fovea, and you can't see color because you don't have any, the cones are not activated. In photopic conditions, it's normal ambient light. This is where most of our vision happens. This is when we can see with our cones. So there's enough light coming in to activate our cones. This would happen in indoor lighting and outdoor lighting. And there's a big difference between indoor and outdoor lighting, but both of them would be considered photopic conditions. Mesopic is a kind of at dawn and dusk, where colors are starting to get a little drained. Your cones are not working as well. They're not getting enough activation, but they're still activated to some degree. And your rods are starting to become activated. This is actually the most dangerous time of day to drive, because your rods are not completely activated and your cones are starting to inactivate. So you actually have some photoreceptors that are offline more than you do when you're in the complete dark or in photopic conditions.